Chapter 6, Viruses and Prions. General characteristics of viruses. They are extremely small. They are filtrable. They are about 20 to about 1,000 nanometers. They are what we call obligatory intracellular parasites. Distinctions from bacteria. Viruses contain DNA or RNA, so they have a single type of nucleic acid, and it can either be DNA or RNA. Contains a protein coat called a capsid, and they are sometimes enclosed by an envelope. They're called obligatory intracellular parasites because they have to be on the inside of a cell to use the cell's machinery. There's no ATP generating mechanism. They transfer the viral nucleic acid to other cells with special equipment. They have few or no enzymes of their own, so they don't have any ribosomes, so they count on the host to do protein synthesis for them. And they're not affected by antibiotics, but are affected by interferon. So here's a picture showing you the sizes of viruses compared to bacteria, which is a lot bigger than a virus. And then, of course, red blood cells are way bigger than a bacteria or a virus. Host range. Species-specific and cell-specific. Most viruses infect only specific types of cells and one host. So, in Chapter 5, at the end, we talked about bacteriophages in the process of transduction. And we said that bacteriophages or phages infect only bacteria. Specificity depends on the receptors on the cell surface. So there are specific host attachment sites and cellular factors. The host range is determined by those specific host attachment site and cellular factors. General categories of viruses are based on the host. So bacteriovirus, bacteriophage, again, the virus detects and attaches to cell wall, fimbriae, or flagella. Animal viruses detects and attacks to receptors in the plasma membrane. Structure of the virus. Again, in order for us to see them, we have to use an electron microscope, and they're about 20 to about 14,000 nanometers. The viron is the extracellular form of the virus. It's the vehicle for transmission, so from host to host travel. The virus cannot replicate or undergo metabolic functions unless it's inside of cells. All virons have the following nucleic acid. The possibilities of nucleic acid are single-stranded or double-stranded, DNA or RNA. So you can have single-stranded DNA or single-stranded RNA, or you can have double-stranded DNA or double-stranded RNA. The RNA or the DNA can be linear, circular, or segmented. So we know for us, ours is linear in eukaryotes. In bacteria, it's circular, and viruses can be anyway. Capsid is a protein coat which consists of capsomeres. So it's composed of capsomeres, and capsomeres are your protein subunits. So here you see this is a capsid, and each one of these little circles are capsomeres. And these capsomeres can be a single protein, or they could be many types of proteins, and you can actually see the nucleic acid inside. The capsid gives the virus its shape, and it helps it to attach to the host cells. So this is how the virus usually travels. So if you look at a door handle, someone sneezes, it puts it on the door handle, then it goes into your hand. So that's how it travels. Some viruses are enclosed by an envelope. So here you see that you have a capsomere and your nucleic acid, but outside of that capsomere is an envelope. So it's found on the outside of the capsid, and it's made of lipids mostly. Some viruses have spikes. Spikes are protein and carbohydrates attached to lipids, and they assist in attachment. So you see here, this black represents the spikes. Okay? The spikes help us to diagnose viruses involved in viral infections. For example, H1N1 or H2N2, that represents the different types of spikes. Examples of animal virions with envelopes is influenza or herpes simplex. Morphology. Remember, morphology is shape. Helical is long, rigid, or flexible rods, so rabies or Ebola. Polyhedral is icosahedron or 20 faces of equilateral triangles and 12 corners. It's many-sided, so polio or adenovirus. Envelope virus can be helical or polyhedral, but it is spherical with an envelope. Complex virus, example of this is bacteriophage, so mostly bacteriophage, and it has a head, a sheath, a tail, a base, and a pin. The head is polyhedral, and the head also contains nucleic acid, and the tail is helical. So let's take a closer look at these morphologies. So helical, you can see those long, rigid, or flexible rods, okay, with the nucleic acid on the inside. So Ebola virus is an example of that. Polyhedral, you have your 20 equilateral triangles with your 12 corners. So you also see that we have nucleic acid in there. Remember, envelope virus, they can either be helical or polyhedral, but with an envelope around them, they will appear to be spherical. And then finally, your complex virus, in which you have your bacteriophage. And remember, you have a head, a sheath, a tail, a base, and a pin. The head, of course, is shaped like icosahedral or polyhedral. Okay, so it has the 20 triangles, do you see that, with the nucleic acid on the inside. And then, of course, the tail portion, you see the helical? Okay, that's the helical portion. Viral taxonomy. In the past, methods were based on symptoms. Now it's based on nucleic acid, the morphology, and the replication strategy. Viruses are classified into families, orders, genera, species, and subspecies. The family names end in viridae. The genus names end in virus. The common names are used for the species, and the subspecies are designated by a number. Table 2 in your book on page 169 has a family of viruses that affect humans. Please review and note for the exam. Viral species are a group of viruses sharing the same genetic information and ecological niche. Okay, so they share the same host and genetic information. So let's take a closer look at an example of the classification. So herpes viridae is the family name. Herpes virus is the genus name. And then the common name is human herpes virus. And then the subspecies is HHV1, HHV2, HHV3. Same thing with Retroviridae. Retroviridae is the family name. The genus name is lentivirus. The common name is human immunodeficiency virus. And then the subspecies is HIV1 and HIV2. Growing bacteriophage. Viruses must be grown in living cells. We can't just put them on media, right? So they have to be the obligate intracellular parasites, so they have to be living in something. So liquid or solid media is used with appropriate bacterial cells. We cannot visualize them in a liquid culture, so we can visualize them on solid media. And on solid media, they can be visualized as plaques. Bacteriophages form plaques on a lawn of bacterial cells. So we make a lawn of bacterial cells. Then the virus is plated on top of that bacteria. The virus then lyses the bacteria, producing a clearance zone called plaques. So what you can see here is those nice little holes that represents the little plaques. Each one of these was a virus. Animal viruses. Animal viruses may be grown three ways. They be grown in living animals, so live animals to study infection, or in embryonic eggs for maintaining growth, or in cell cultures. Cell cultures is the preferred method. Animal cells are treated with enzymes and nutrients are formed to form a monolayer. So we treat the animal cells with enzymes and nutrients, and then they form a monolayer. From there, we take a look at the cytopathic effect of an infected monolayer. So we're going to infect the monolayer with a virus, and those transformed cells are then going to be infected. No monolayer is going to be formed, but instead what ends up happening is a tumor is formed. So we could take a tissue, treat it with enzymes to create them, to separate them to make cells, and then those cells are going to be grown to monolayers. Right? So this is our cell culture. And these cell cultures may be called continuous cell lines because they may be maintained indefinitely. However, if I infect this monolayer with a virus, they're no longer going to grow in a monolayer. They're going to grow as tumors, in which they're going to grow on top of one another. Here ends Part A of Chapter